2 toxins should be secreted by a type 2 secretion system. That's not the case. They're just numbered because that's just an easy way to categorize them. They could easily be numbered category A, B, C, D. Okay, so don't get them confused in terms of toxins and secretory systems. And I will try to make that abundantly clear throughout the lecture. Okay, so we're going to be talking about 2 different types of toxins. And this is a very easy concept to understand. So there's exotoxins and endotoxins. Exotoxins are ones that are secreted outside of the bacterial cell. So they're exotoxins. And they kill host cells, they release nutrients so the microbes can acquire those nutrients. And a lot of times this secretion is tied to attachment. So attachment is a prerequisite for secretion of a particular toxin. And that makes sense because if you're just secreting it out randomly in the milieu, then you have a lot less chance of that toxin getting to where it needs to be, which is in contact with the host cell. So if you are already in contact with the host cell and then you secrete your toxin, it will be much more effective because there's not a dilution factor that's happening. It's in close proximity to where it needs to be. Endotoxins are not secreted. These are very different than exotoxins. It's part of the bacteria itself. Okay, so it's endo, it's inside, well, not inside, it's on the surface of the cell. But it's something that's not secreted, okay? We're going to talk about it's what it is in detail in a minute, but if you remember when we talked about the structure of the cell envelope and we talked about lipopolysaccharide in this case is our endotoxin. So in certain literature, particularly medical literature, you will see that LPS is actually referred to as endotoxin. So why is it so toxic? Well, it's something that only gram-negative bacteria make. So hosts, like humans, have evolved to recognize it as foreign. Okay, so it's a pathogen-associated molecular pattern, and it triggers host defense responses. Because if you are invaded by a bacterium and your body senses that there's LPS in large amounts inside of you all of a sudden, then of a particularly pathogenic kind, it will mount a defense response. Sometimes that those checks and balances of defense responses trying to kill bacteria get a little bit out of whack, okay? And so the host will mount too much of a defense response and a sort of an unchecked defense response. And then you get into sort of an autoimmune type of situation where you go into endotoxic shock because you're mounting this defense response in response to this endotoxin, and you're causing a lot of swelling and edema in your body that is an undesired side effect, and you can go into endotoxic shock. Okay? All right, so this is just a diagram of our exotoxins. So these are secreted outside of the cell as shown by the little red dots. And endotoxin is something that is part of the bacterial cell, okay? And endotoxins can be released in large amounts when phagocytes or other immune cells degrade the bacteria. And then suddenly you have all this free-floating endotoxin or LPS floating in the bloodstream or in the gut or wherever it has been released. Okay. So, like I said, they are secreted generally following attachment, and these exotoxins fall into 5 broad categories that we're going to be talking about. These include cell membrane disruptors, so things that poke holes in membranes of host cells. Okay, that's a very conceptually easy thing to understand. There are some that block protein synthesis by messing with the ribosomes. There are some that block secondary messenger pathways, so signal transduction, like G protein signaling, we're going to talk about. Some are superantigens that overactivate the immune system, and some are actually proteases that are involved in cleaving host proteins and degrading them. Okay, so we are going to focus on the first 3 categories, just for the sake of time. But I did want you to know that there are these 5 classifications of exotoxins. Then I put this table in here from your textbook. I'm not asking you to memorize every single thing on the table, but I wanted to just give you an idea of the different categories and what bacteria are producing what toxins that belong into each of these categories. Okay, and you will see toxins that I'm going to mention in detail, like cholera toxin, diphtheria toxin, hemolysin. So I do expect you to be responsible for those, but not ones that I'm not mentioning, like pneumolysin or something like that. Okay. So the first category is damaging cell membranes, and you can see that a lot of these toxins have lysin on the end of them in their names, and that just means that they're lysing host cells. It's very descriptive. Then we're talking about ones that inhibit protein synthesis. We're going to talk about Shiga toxin and diphtheria toxin there that are produced by specific bacteria. And then the ones that activate secondary messenger pathways, our example there is going to be cholera toxin. And then these ones we're not going to take into much detail for due to time constraints, but we have ones that activate the immune response. Something that you might be familiar with is toxic shock syndrome caused by Staphylococcus aureus, and then also proteases that are involved in tetanus. Tetanus toxin is a protease. Okay, so these are the three categories that we're going to talk about. We're going to talk about the ones that damage cellular membranes, and how they do that is they form pores in their host cell. So they are these toxins that are very good at forming pores. I'm just going to skip to the next slide. So alpha toxin or hemolysin toxin from Staphylococcus aureus is sort of the prototypical toxin, this alpha hemolysin, that fits into that first category. Okay, so our membrane destroyers. And so you can see here that this three-dimensional reconstruction of the structure of this protein, it looks like a barrel, right? So it's a thing, it's like something that will insert into the membrane, and once you disrupt the membrane in such a way, it will cause the host cell to lyse. Okay, so the target cell here are red blood cells, so hemolysin, that's why you get the word hemolysin. And it's actually a very good diagnostic tool if you're in a clinical diagnostic laboratory where they're trying to determine if the Staphylococcus aureus that has been isolated from a patient has hemolytic capability or not. So that's one of the criteria for categorizing different Staph aureus strains. And so they simply plate it on media that contains sheep blood cells. And if you get this halo around it, that you can see over here, that means that bacterial colonies secreting hemolysin that is breaking down those blood cells in that vicinity through diffusion. Okay? And there are many different types of lytic proteins produced by other bacteria, but our example is alpha hemolysin from Staph aureus. Okay, so Shiga toxin, this is our example of our category 2 toxin, where we're talking about disrupting protein synthesis by messing around with ribosome function in the host cell. Okay, so this is produced by Shigella flexneri. That's where it gets the term Shiga toxin, because it's short for Shigella. And also E. coli. So Shigella and E. coli are very closely related to one another, and so they can participate in a lot of gene exchange. And some of that has to do with Shiga toxin. Okay, so if you're E. coli 0157H7 and you have acquired this ability to produce Shiga toxin, then you are extremely virulent, and you cause a lot of diarrhea in people who ingest this. It's also been nicknamed the Jack-in-the-Box microbe because in the 90s, it was found in ground beef that was served at Jack-in-the-Box restaurants and caused an outbreak of E. coli and Shigella diarrheal diseases associated with that particular restaurant. So how it works is it destroys the 28S rRNA that's found in the ribosomes, okay? And it's also, we're going to talk about it, it's an AB toxin. So AB toxin just means, it's very simple, it means you have an A part and a B part. The B part is sort of a chaperone for the A part, and you can see that this B part is associated with other B parts, with 5 B parts, okay? And it shuttles the A part, which is the actual toxin, to the host, and then the toxin, or the A part, is secreted into the cell, and that is what goes and destroys the 28S ribosomal RNA in the case of Shiga toxin. Okay? So just again, A-B toxins just mean that there's an A part and a B part. So there are a lot of toxins that have this bipartite structure. They need a chaperone to bring the toxic unit to the cell and inject it in, okay? All right, so I'm just going to, I always don't know which one I should do first. Okay, so I'll just skip to this A-B toxin thing again, just to show you that A subunit, B subunit, the A subunit is the toxic part, okay? So now I'm going to go back to ADB ribosylating toxins. So this is just a mechanism of how a toxin functions, and this is what we're talking about in terms of the category 3 toxins are examples of the cholera toxin. But I wanted to show you that ADB ribosylating toxins can also belong to category 2, okay? And all it means is that you are ribosylating something, okay? So you are shuttling an actual ribose sugar to a target, okay? In the case of the diphtheria toxin, you're ribosylating elongation factor 2, okay? Which blocks ribosome function. That's what makes it shift into our category 2, where we're talking about blocking ribosome function and protein synthesis, okay? But the mechanism is through ADB ribosylation, okay? So we are adding a ribose to elongation factor 2, which screws up protein synthesis, and that's why it's in category 2. Category 3, or cholera toxin, which is made by Vibrio cholera, which is the causal agent of cholera, it ribosylates through this ADB ribosylation to overactivate adenylate cyclase. And adenylate cyclase is interacting with G protein signaling, so that's why it fits into our category 3 of screwing around with secondary messenger pathways, okay? And then I'm just going to bring this back here to talk about these AB toxins, okay? The A subunit has the toxic activity, and in the case of cholera toxin, that A subunit is the ADB ribosyltransferase. So that's what categorizes it as an ADB ribosylating toxin. And diphtheria toxin also has this A-B subunit partnership. Okay, so I just want to reiterate that A-B toxin can be found in lots of different categories, and it just means that there's a partner that shuttles another partner, and that the A part is the toxic part. Okay, so now we're going to talk about cholera. It's very interesting. It's a waterborne disease, so it's caused by Vibrio cholera. If you remember, that can cause a problem in addition to the infection, and maybe even worse than the infection they already had. Okay, so now we're moving along. We left toxins, right? We're going to move into secretion pathways. And some toxins are secreted through these secretion pathways, right? So it's a way to deliver specific proteins to host cells. And we're going to talk about that in the context of toxins, but also other things, okay? And sometimes these proteins, they're not destined to kill the cell, but to just mess with the cell a little bit, okay? So they can interfere with signaling within the cell. They can dampen host immune responses. So I just talked about pathogen-associated molecular patterns triggering the innate immune response. So that's called PTI, or PAMP-triggered immunity, okay? So that's something that just happens because the bacteria can't help it, right? They have LPS, and the host is going to respond to that. So how do they answer back? Well, they can answer back by secreting effectors into the cell that dampen that response. So it's like a constant arms race between the pathogen and the host. So the host has evolved to recognize these PAMPs, and then the bacteria initiates PAMP-triggered immunity, and then it's like, okay, wait a minute, but I don't want to get killed by this innate immune response, so I'm going to secrete an effector in there that will dampen that right away. And so then the host can sometimes create a protein that will dampen that effector, and then so on and so forth. There's this arms race that goes on with this battle between pathogens and their hosts. I will mention here that a lot of our secretory systems look like other things we've already studied that are innocuous, okay? So an example of that would be our type 4 pili. So we know that those are just involved in switching motility, and they're not involved in secreting toxins. But we can see that there are secretory systems, like the type 2 secretion system, that when you look at them structurally, they look a lot like the type 4 pilus. And it's involved in extension and retraction in the same sort of manner. The same with the type 3 secretion system that we're going to look at. It looks a lot like the flagella assembly with the motor and everything and the way that it's made. And so it's like this co-evolution or convergent evolution of two things that have similar structure, but one has evolved to either be a type 2 secretion system or a type 4 pilus or a flagella or a type 3 secretion system. Okay, so there are 4 secretion systems we're going to talk about. Right now, we're up to, I think, there might be a new type 7 secretion system. They keep adding new ones that have been discovered. But we're just going to talk about 4, type 1 through type 4. Okay, type 1 secretion system can just be classified as a general secretory pathway. So a lot of things are secreted through that. Not necessarily toxins, just things that need to get out of the cell. Type 2 secretion system, it looks, like I said, like a type 4 pilus, and the way it works is through extension and retraction of the piston through the type 2 secretion system, very similar to the extension and retraction of the type 4 pili. But in this case, a protein gets inserted and it gets jammed up to the system. Okay, so instead of just the pilus going back and forth for motility, a protein is in there and it gets shot out of the cell. Type 3 secretion system is very cool. It's very well studied in lots of gram-negative organisms. It looks like a needle. Okay, so it looks like a molecular syringe that these bacteria are injecting things into their hosts. And the type 4 secretion system, also very cool. So it really looks like conjugation machinery, or the sex pili that we talked about when bacteria are mating with one another. Okay? In this case, we're going to talk about it in terms of cross-kingdom DNA transfer. So a bacteria moving its own DNA into a plant host. So cross-kingdom DNA transfer. All right, so let's talk about type 1 secretion, or the seq system for short. So it's general secretory pathway, like I talked about. It has 3 different parts. So you have this ATP binding cassette protein that is providing the energy. It has a periplasmic protein that stretches across the periplasm, which is this rod-shaped thing here. And then you have an outer membrane channel protein. So you need all 3 of those parts to get things out of the cell. And one of our examples of something secreted by the type 1 secretion system is the hemolysin, or the alpha hemolysin that we talked about when we were talking about toxins. Okay? So alpha hemolysin is secreted through the type 1 secretion system. And generally, if you're looking at the genomes of bacteria, you will find this type 1 secretion system, the genes that encode all these proteins in this channel over here, right next to the alpha hemolysin, okay? And sometimes in the same operon. So that makes sense that they are transcribing and translating themselves along with their secretory system. Okay, type 2 secretion. Again, very similar to our type 4 pili that I was talking about, except in this case it's modified for secreting proteins rather than for mediating motility. So again, you have this polymerization of these pilin subunits, these blue ovals here, and through the polymerization and depolymerization process, you can shove in a protein in there, and then as it polymerizes, it shoots the protein out of the cell. And when it depolymerizes, you can have another protein inserted in there. As it polymerizes, it shoots it back out of the cell. So it can extend and retract just like the type 4 pilus. An example of something secreted by the type 2 secretion system is cholera toxin. Okay, so the type 3 secretion system, we know a lot about this because a lot of gram-negative bacteria have this capability, including E. coli and its close relatives, Salmonella and Yersinia. So Yersinia pestis is the causal agent of the bubonic plague, like I talked about, and E. coli and Salmonella are involved in gastrointestinal diseases. Pseudomonas aeruginosa, again, it colonizes the lungs of cystic fibrosis patients, and Pseudomonas syringae is a very adept plant pathogen. So all of these are gram-negatives, not necessarily closely related to one another, but they all have this type 3 secretion system. So there are many researchers on this planet who are either mammalian or human microbiologists, as well as plant microbiologists, looking at the type 3 secretion system. So there's a huge body of literature on this secretion system. And what we know is that it looks very similar to the flagellar motor, and also the flagella itself. Okay, so you have this flagellar motor, very similar to what the type 3 secretion motor looks like. And if you are to burst open a cell, and you can isolate these type 3 secretory components. Okay, so you can see here in panel B, we have all of these type 3 secretion components that have been isolated from the cell and then visualized by electron microscopy, and they look like little needles, like little syringes. Okay, and it's a one-step injection. It requires attachment to the host cell. So this is a very good way of getting something into your host without allowing it to float out or have to attach to the host cell. You're bypassing that need. Okay, so you're just like, yourself is poking it into the host and then secreting what we call effectors. Okay, so there are a lot of type 3 secretion effectors, and that's just a catch-all term for proteins that are secreted by the type 3 secretion system. Okay, so our example is Salmonella in this case, and it has, in this example, over 13 effectors that is secreting into the host, and it's initiated by tight attachment to the host cell. Okay, so this is a gut epithelial cell. This Salmonella bacterium that is propelling itself by flagella attaches to this gut epithelial cell, which causes the host to engulf the bacteria, so it causes a conformational change in that epithelial cell. And it starts secreting what we call these type 3 effectors into the cell, these little blue dots here. And they have a lot of different jobs. One of them is to immediately suppress host immunity. So when I talked about PAMP-triggered immunity, this would be something that these effectors go and suppress that immediately. So that gives that bacteria a better chance of survival in that host. Okay, we have an animation to show this.
Many bacterial pathogens, including Salmonella enterica, use tiny molecular syringes embedded in their membranes to inject proteins directly into the host cell's cytoplasm. This mechanism of delivery is called type 3 secretion. In the case of Salmonella, type 3 secretion also allows the bacterium to invade a eukaryotic host cell and become an intracellular parasite. The type 3 secretion system includes a variety of proteins arranged into a microscopic needle. The base of the needle complex spans the inner and outer membranes of the bacterium. The thin needle projecting away from the cell is made up of multiple subunits of a single protein. The tip of the needle must make contact with a host cell to initiate secretion. Upon contact with the host cell, a signal is transmitted through the needle, triggering the creation of a pore complex in the host cell's plasma membrane. Salmonella then uses its type 3 protein secretion system to deliver a cocktail of at least 13 different protein toxins, called effector proteins, directly into the cytosol of host cells. Inside the bacterium, chaperone proteins bind to the effector proteins, delivering them in a loosely folded form to the base of the needle. An ATPase triggers the release of the effector proteins and helps them to unfold and begin their journey through the narrow channel in the needle. Direct delivery to the cytoplasm of the host cell is a good idea because it eliminates the dilution that happens when a toxin is secreted into media. Inside cells in the gut, these effector proteins interfere with signal transduction cascades and modulate the host response. One mission of these effectors is to induce cytoskeletal rearrangements that cause ruffling of the host cell's membrane around the microbe, leading to bacterial engulfment. Once Salmonella enters a cell, it finds itself in a vacuole called a phagosome. In the normal course of events, a lysosome would then fuse with the phagosome and release its contents of digestive enzymes in an effort to kill the invader. Salmonella, however, uses another type 3 secretion system to inject proteins that alter vesicle trafficking, thereby decreasing phagosome-lysosome fusion so that the intracellular bacteria are spared. By using its type 3 secretion systems, Salmonella subverts the normal function of host cells and causes an inflammatory response leading to diarrhea. The genes encoding the type 3 systems are located within pathogenicity islands on the bacterial chromosome. Pathogenicity islands are typically inherited from other microbial sources. Salmonella has acquired at least 5 and as many as 10 pathogenicity islands that are absent from related but harmless E. coli strains.
Right, so I forgot to mention that a lot of times you can find these type 3 secretion system operons in pathogenicity islands. And so they're readily exchanged from bacterial species to another bacterial species. Okay, so that was type 3 secretion. Now you have a lot of knowledge for which you can do your own graduate studies in type 3 secretion. Yes?
What's the difference between like category 1, 2, 3 and the secretion signal 1, 2, 3?
So it's just numbering of systems. So they don't have anything to do with one another. So I mentioned that in the beginning of class where category 1, 2, 3, 4 toxins doesn't match up with type 1, type 2, type 3, type 4. Category of secretion systems, we could have easily named it category A, B, C, D in type 1, type 2, type 3, type 4. They have nothing to do with one another. It's just a way of organizing things. Yeah. I mean, they do have something to do with one another in the fact that you're secreting toxins through these different secretory systems, but the naming is no cross-reference there. Okay. So now we're going to talk about type 4 secretion. And like I mentioned, it looks very similar to the conjugation pilus that is needed for the mating process in bacteria, so like the sex pilus that we talked about. But in this case, it can secrete both DNA and proteins. It's not just the DNA secretory system. It can extend and retract as well. And it can either target proteins or DNA outside of the cell or directly into the target cell. Okay? So an example of this would be the pertussis toxin, which is produced by Bordetella pertussis, which is the causal agent of whooping cough. And it's secreted through a type 4 secretion system. And it's an aging toxin. As you can see, it has the A and B part. But how we're going to talk about it is in this really cool plant-associated bacterium called Agrobacterium tumefaciens. So in this case, it's not secreting proteins, it's secreting DNA. And it's moving that DNA from the bacteria into the plant host nucleus. Okay, so that is no easy feat. It has to cross a plant cell wall, which is made of cellulose and lignin and other things. Plant cell membrane, which is not a barrier that just lets anything freely move past it. Then it has to get that DNA into the through the nuclear membrane into the nucleus, and that DNA integrates into the plant cell chromosome. And then what happens is you get these galls. Okay, so the bacteria, through this DNA transfer, are inducing production of these galls. And obviously that's a problem for a plant. You have this big growth on the side of you. And what happens is it disrupts the hormones pathways and makes the plant make more of the plant hormones. And you have uncontrolled growth, or like a plant cancer, of these plant cells. And you get these like warty-like structures on the side of a plant. And if it goes unchecked, you end up with something like that on the side of a tree. Okay? So this is an example of cross-kingdom DNA transfer. How it works is that you have these bacteria, these Agrobacterium tumefaciens, that really like to live in the soil. They're very good soil dwellers. And when they come into contact with a plant root, they can engage with that plant root and colonize it. And in doing so, it will adhere to the plant cell, and then through a series of eliciting events, it will then secrete its DNA through mediated by a plasmid called the tumor-inducing plasmid into the plant cell. Okay. Now the whole plasmid doesn't move over like when we talked about conjugation and the S factor. Only a piece of the plasmid, and that piece is called the tDNA or the transfer DNA. So that's the DNA that's being transferred. And what's the deal with the tDNA? Well, the tDNA encodes plant hormones. Okay, so this bacteria has these genes that encode plant hormones. And so it encodes auxin and cytokinin. And so once that gets integrated into the plant cell nucleus and into the plant cell chromosome, it starts making plant hormones in an uncontrolled way. So plant cells, they have evolved to control the production of their hormones when they need them, but this bacteria has hijacked the system and said, you're going to make these hormones all the time. And what happens is you get hyperplasia and hypertrophy, which is uncontrolled cell growth and uncontrolled cell division. So you get big giant cells that divide a lot, and then you get what we call a gall. Okay? So basically the bacteria are inhabiting these galls, and so they've told the plant to make a little house for it. And so it's this niche that it can only occupy, and so it's created this very exclusive neighborhood for itself where only it can live. And how does that happen? Okay? So in this tDNA, there's also genes that encode specific or special amino acids that only this agrobacterium strain can use as food or a carbon source. And so it makes a house for itself, and then it makes this exclusive niche for itself by secreting these special amino acids that only it can utilize. And so it lives in this house, and it tells the plant to make its food and please feed me on a regular basis, and I'm going to exclude all others. And so that is how that system works. And it's a very good example of cross-kingdom DNA transfer, which is very rare. Okay, now I'm going to talk about this. How much more time do I have? Oh, plenty of time, 9 minutes. So I just talked about this really cool thing that we've studied, which is moving DNA from a bacteria to a plant cell. Of course, humans, when we find something interesting, we always figure out a way where we can use it for ourselves. And so we've decided that this is a great way to create transgenic plants. Okay? So this is just an overview of how tDNA synthesis works in nature when we're talking about crown gall disease. I just want to talk about that one more time. So the yellow region here is the tDNA. There's a left border and a right border. These are just conserved nucleic acid DNA arrangements of base pairs that are recognized as the left border and the right border. Inside of that are the auxin, the cytokinin, and the genes that encode those special amino acids. So those are the things that get moved over. We have discovered that you can remove those genes, so auxin, cytokinin, OP biosynthesis, keep the left DNA and the right DNA borders intact, and whatever gene you put inside of there will also move. And so if you want to make a transgenic plant that's producing lots of beta-carotene, for example, you can just swap it out, those auxin, cytokinin, and amino acid biosynthesis genes, with a gene that encodes a carotenoid. And then you move that into your plant, it gets into the plant chromosome, starts making loads of beta-carotene. Okay? So this is how we make transgenic plants, for the most part, in the scientific community. Okay? So it's based on this TI plasmid, or tumor-inducing plasmid, that this T region or tDNA region is transferred into the plant chromosome. And we can swap out whatever we want, with some limits, in that between the left border and the right border. Okay, so this is called agro-mediated plant transformation. So any plant transformation facility on any university campus, much like we have here at UCR, utilizes agrobacterium to transform plants. And then you can get that plant to express a gene of interest, whatever it is you're looking at. Okay? And sometimes can be used to transform fungi as well. Now, we have to disarm the TI plasmid, which means removing the auxin and the cytokinin and those amino acid genes I was talking about. And then we also, probably more detail than you need to know, but you have to split the TI plasmid into two because it's so big. And so moving big pieces of plasmid DNA around is difficult. So you just split it in two, and so you have to have two plasmids. You have this one carrying your gene of interest, and then you have what's called a helper plasmid. And the helper plasmid is helping the DNA move from the bacterium to the plant. And in a nutshell, this is how it works. Okay, so you have your agrobacterium that you have cloned in your gene of interest into your TI plasmid, and then you expose it to your plant cells. These are generally protoplasts or individual cells of your plant. And then you have this transformation event that occurs so that tDNA moves over, inserts into your plant cell, and then you can select for that because also in that tDNA you have put in some sort of a selection marker that allows for only the plants that have been transformed to grow and kills all the other ones that haven't been transformed. So then you have to nurse them along with these little petri dishes until they finally turn into little plantlets, and then they grow into large plants, and then you can test them for whatever phenotype you're looking for. So if you think that you are having them produce a lot of beta-carotene, you can test beta-carotene levels in a transgenic versus a non-transgenic form, or whatever it is. A lot of defense-related genes are moved into plants this way, so moving a defense-related gene and having it expressed in all tissues might make that plant more resistant to pathogen attack, for example. So that's how we make a lot of transgenic plants, and it's a very efficient way, depending on your plant species. Okay, so other fun virulence factors that we're going to talk about, we have siderophores, hormones, and ice nucleation. So siderophores are iron-grabbing chemicals that are secreted by bacteria. So bacteria, well, any living organism, it always needs iron. Okay, and so iron is generally in limited supply, say in host tissues. So there's this constant race between who's going to get the iron. So bacteria have evolved to secrete siderophores that bind iron, and then they have their own siderophore receptor protein that then brings that siderophore containing the iron back into the cell. So it can grab it from other bacteria in the environment, or also grab it before the host can utilize it. And so this is an iron sequestration mechanism. We're going to talk about ice nucleation, and because we already talked about hormones as being a virulence factor, so agrobacterium uses plant hormones as a virulence factor. Ice nucleation is something fun that was originally discovered by Professor Lindau at UC Berkeley, who was working in this system where he saw that in the field, certain plants were not as susceptible to frost damage as plants in a different field. And so he was wondering, okay, the temperature has dropped to below freezing. Why are these plants not susceptible to frost damage? And he discovered that there's actually a bacteria that was associated with these plants that was protecting the plants from frost damage. And that the other plants had other bacteria on them that were serving as ice nuclei. So anytime ice is going to form from a liquid to a solid, you need something for that ice lattice to form around, and that's an ice nucleus, okay? That can be a piece of dust, that can be anything, and it can also be a bacterium. And so they found these ice nucleating bacteria that can cause things to freeze at higher temperatures than would a bacteria that does not have ice nucleating properties. So, for example, these two plants were crushed. One would contain ice nucleating bacteria, others didn't, and they were subjected to just above freezing temperatures. And you can see that the plant on the right caused that liquid in that test tube to freeze, and that's because it had a high concentration of ice nucleating proteins. And so from that, there were some biocontrol applications that were derived based on spraying plants with bacteria that didn't have ice nucleating properties. So that would prevent colonization of the plants with bacteria that had ice nucleating properties. And then another fun thing, if you go to Big Bear, they make a lot of their own fake snow up there. So this has been utilized to make fake snow. Okay, so this is a positive thing from ice-nucleating bacteria. So when they're trying to create snow in temperatures that might not be quite freezing, and they're making artificial snow, a lot of these snow blowers use this product called SnowMax, and it's actually a bacterium that's serving as an ice-nucleating, it's providing its ice-nucleating properties to that snow machine and spraying that artificial snow. And then frostban is that one where it's an ice minus bacteria that we use for biocontrol in plants.